Welcome everyone. Today we are going to learn about hydropathy plot. But before I jump to the topic, I want to mention that this hydropathy plot is related with integral membrane protein. I have already made a separate video on integral membrane protein, and in that video I have mentioned that integral membrane protein contains three domains. One is exoplasmic domain, that is outside of the cell. Another is transmembrane domain that spans the lipid bilayer or traverses the lipid bilayer, and the cytosolic domain that presents the cytosol. Now, the transmembrane domain of integral membrane protein is predominantly alpha helices. Now, single alpha helix can traverse the lipid bilayer. This is one example. This is called single pass integral membrane protein. Another example is there could be multiple alpha helices like this, and we call it multipass transmembrane protein or integral membrane protein. Now, what is hydropathy plot? Hydropathy plot helps to identify these alpha helices present in the integral membrane protein. So let's talk about hydropathy plot. Now it helps to localize potential alpha helices of transmembrane domain of integral membrane protein. About 20 to 30 amino acids hydrophobic in nature. Remember this portion transmembrane domain of the integral membrane protein should be hydrophobic in nature because it traverses the lipid bilayer, and lipid is hydrophobic in nature, we know. So this amino acid should be hydrophobic in nature also because they can then interact with the hydrophobic fatty acid chains with van der Waals interactions. So about 20 to 30 amino acids are hydrophobic in nature, long enough to span the lipid bilayer. Now, by convention, hydrophobic amino acids are assigned positive value, and hydrophilic amino acids are assigned negative values, and this is called hydropathy index. As you can see, this is the in the y-axis there is a hydropathy index, and here is the number of amino acids. Now, basically, this calculation actually made for segments of fixed size, usually 10 to 20 amino acids in the protein. So in case of glycophodin, scientists have found one positive peak. That means there is one alpha helix or single alpha helix. So it's a single pass transmembrane protein. And in case of bacteriorhodopsins, there are 7 peaks. It has been found in the positive value. So that means they are hydrophobic in nature. So they are actually 7 alpha helices that actually traverse the transmembrane protein. So this is called hydropathy index. Conventionally, the hydrophobic amino acids are assigned positive and hydrophilic amino acids negative. Now, if presence of 10 to 20 amino acids or 20 to 30 amino acids are enough to traverse the or span the lipid bilayer, and it has been found that by this hydropathy plot we can recognize the potential alpha helices. Now, it cannot identify by the hydropathy plot, you cannot identify the transmembrane domain of beta barrel. Now, what is beta barrel? Beta barrel is made up of this beta barrel proteins, actually integral membrane proteins, but the transmembrane segments here is made up of beta strands, another secondary structure, unlike the alpha helices. Okay, so it cannot identify by hydropathy plot, you cannot identify the transmembrane domain of beta barrel, as only 10 amino acids or fewer are sufficient to span a lipid bilayer. Now let's see. So this is an example of glycophodin. Glycophodin make actually dimer. It is a biological unit of dimer, I have already mentioned, and scientists found one peak in the single subunit of glycophodin. Each subunit contain one peak. And bacteriorhodopsins, one example of multiplus or 7 transmembrane protein. Here you can find 7 peaks of alpha helices. But in case of podin, see this structure is beta strands. So there are multiple beta strands makes the beta barrel or pod-like structure is called podin. And if you see the hydropathic plot, you will not find any prominent peak of alpha helices because only 10 amino acids or fewer amino acids are enough to span the lipid bilayer, but just by these 10 amino acids you cannot identify the alpha helices. So it is not possible to identify the beta barrel structure by this way or hydropathy, but you can identify the alpha helices of integral membrane protein. So this is the significance of hydropathy plot. Hope this video will help you. Thank you for watching, and always keep learning.